Good day, students. Today's practical session is titled Stem Anatomy of a Dicot plant. This is an example of a dicot plant, and the name is Tridus procumbens. Now, the aim of this experiment is to determine the anatomy of a stem of a dicot plant. Now, materials that we're going to be using, we have a series of ethanol, 50 to 100%. We have saffron, our stain. We have glycerol. We have nail varnish. We have a petri dish containing distilled water, sodium hypochlorite. Now, to get the stem of this dicot plant, we'll be using this styrofoam material. So we'll cut. This is because we want to mimic using a microtome machine. So you cut and put your stem in between the styrofoam material, and you make several cuttings into the still water. So make a block so that we can mimic using a microtome machine, which is used for sectioning of tissues. So we'll make several cuttings into the still water. But you must make sure that your cuttings are very, very slim. We'll make a very thin section so that you'll be able to, because if it's not a thin section, light will not be able to pass through if it's too thick. So you make several cuttings into the still water. You can see. So this is transverse section of a dicot plant. You pick your slide. You have cut it into water first to hydrate your plant. You want to hydrate your sample. That's why that's the first stage. You put it in water to hydrate your cell. Then you put a drop of sodium hypochlorite and leave it for about 10 seconds. Now, sodium hypochlorite will decolorize the cell so that when you are staining, the plant organelles will pick the right color from our stain. So after you have decolorized, you add water again to remove the effect of the decolorizer, the hypo. You add water to it to remove the effect of the decolorizer. Then you now have to add in the cells to enhance proper staining. We have dehydrated the cell. Now we have hydrated the cell, so we want to dehydrate the cell with grains of alcohol. So the first alcohol we'll be using is 50% alcohol. We are dehydrating with grains of alcohol so that we don't want to use 100% at once, so that we will not break the cell. So we'll start with 50% alcohol to remove the water that we have put there. We are dehydrating. You put a drop of alcohol and you leave it for like 5 seconds. That's 50% alcohol. So you now put also 70% alcohol. Just a drop, leave it for a few seconds. You can drain the excess. Then you put 100% alcohol. This dehydration is for us to keep the plant in a good shape, in a good form, so that when you are staining, it will pick the stain very well. Now after this, we'll put our choice stain, Saffron O, 1% Saffron O. You put a drop, and you allow it to stay for like 30 seconds. After 30 seconds, you put a drop of Glycerol. So allow it to stay for about 30 seconds. So after 30 seconds, we add of staining with saffron, we add a drop of 1 to 2 drops of glycerol. Now the saffron will enhance proper staining, then glycerol will enhance easy view or easy visibility or clearer view when you check under the microscope. Then we now cover with cover slide gently from one side to prevent trapping air bubbles. So you put the cover slip from one side gently, then you lower the cover slip gently to prevent trapping of air bubbles. So your stain must cover the whole part of the cover slip. If not, you will be having air bubbles in your preparation. So stain must cover the whole surface of. So after this preparation, we now rinse the edge, or we seal the edge with nail polish so that our sample will not be dehydrated. We seal the edge with nail polish so that if we are going to be checking our samples, it will still be, this will be a semi-permanent preparation that can stay for months, for weeks and few months. Then now after this, now we'll go and check under the microscope. So we'll check under ×10 objective and first ×4 objective for scanning. Then we'll now check ×10 objective to see a bigger view, which is so after that we are going to put the picture of the video. Thank you. Thank you for watching. If you find this video useful, please kindly like and share and subscribe. God bless you.